Again, today I'm going to be doing life sciences again, but today we are focusing on meiosis. Now, on meiosis, these are the things we have to know: the structure of the cell. You have to know what are the structures or on the cell, right? You have to know what's the importance of meiosis. You have to know abnormal meiosis. Okay, now the first thing with every topic you do, just refer to your marking guideline. Please guys, when you are dealing with life sciences, refer to your marking guideline. If there is anything or answers in the marking guideline, use those. Don't use anything else. Use those, because those are the answers that are going to be used to mark your sheet. So now, meiosis, right? You have to know the definition of meiosis, right? So meiosis is a division whereby gametes are formed, right? That forms haploid gametes, right? It's a division of a diploid cell to form a haploid cell, right? So that's your meiosis. You can define it in any way, but always include the keywords, right? Now, as we said, the first part, we have to know the structure of a cell, right? Now, on your cell, you have your cytoplasm, you have your nucleoplasm. Remember, you have to be able to differentiate between the two, right? Differentiate, please. Differentiate between the two. Plasm just tells you this is a fluid, right? These are fluids, but one is in the nucleus, but one is in the cell, right? Okay, now you have to know your cell wall and membrane. Do not confuse these two. They are not one thing, but they are two different things, right? One is your cell wall, two is your cell membrane. And you have to know your nuclear membrane. Now remember, on your nuclear membrane we have small holes which the mRNA uses to exit the nucleus, right? Those holes are called your nuclear pores. Nuclear pores. Now inside the nucleus we also have your nucleolus, right? It's the smallest. It's like a ball inside the nucleus. So that's your nucleolus, right? It's your nucleolus. Now you have to also know the nucleus itself, right? The nucleus itself is inside the cell, right? It's inside the cell. Now the other parts I want us to know, these are just additions, right? But I want us to also know your ribosomes, right? You have to differentiate between your ribosomes and mitochondria and nucleus. Be careful of those three parts. So I just want us to know these two, ribosomes and mitochondria. And remember, one is mitochondrion, but many is mitochondria. Now the other part is your chromatin network. You have to know it's made up of what? Chromosomes, right? Chromosomes. That's your chromatin network, but the whole structure is made up of what? DNA. Now the next part, you have to know the structure of a chromosome itself, a chromosome on its own. So you have to know this part. You have to know a chromosome has a chromatid and a centromere, which joins two chromatids to make a what? A chromosomes. Now we also have to know that chromosomes in pairs are called homologous chromosomes, but chromosomes during crossing over are called epivalent. And then this part on a chromosome is called your chiasma. The part where the chromosomes join or meet during crossing over is called your chiasma, right? If there are many, it's chiasmata, right? Now that's the part where chromosomes meet during crossing over. So that's the other word, crossing over, right? We'll deal with that later on. So now you also have to know the importance of meiosis, right? You have to know the importance of meiosis. Now, these are the first ones, right? Remember, we have the importance of meiosis in your marking guidelines, so use those. So in your guidelines, it says the first function or importance of meiosis is for the production of haploid gametes. Now remember, meiosis has a halving effect, so which means it halves 46 chromosomes to 23 chromosomes, right? Okay, now the next importance is the halving effect of meiosis, right? The halving effect of meiosis, this what we talked about just a minute ago, a second ago, right? That it halves the 46 chromosomes to 23 chromosomes. And remember, this is haploid, right? And this is diploid. Now, this tells you that all gametes are haploid. All gametes are haploid, and we spoke about gametes when we were dealing with human reproduction. We said gametes are your sperm cell and ovule, right? Refrain from using egg cell. Now, ovule. So now then the importance continues to say the halving effect of meiosis overcomes. You can say overcomes or neutralizes, right? Neutralizes. So it neutralizes or overcomes the doubling effect of fertilization. Now this is the doubling effect of a fertilization. Remember, I have 46 chromosomes. My girlfriend has 46 chromosomes, right? Or let's say my wife, né? So now I have 46 chromosomes, but my gametes are made up of 23 chromosomes, which is haploid, or half the chromosome number, right? Now, my girlfriend also has 23 chromosomes, which is also haploid, half the chromosome number. Now, this is where the doubling effect takes place. 23 plus 23 is equals to your 46 chromosomes. Now, this shows that 23 multiplied by 2, so that's the doubling effect, is equals to 46. So this is the normal human chromosome number. So this importance is to maintain a constant chromosome number from generation to another. So me and my wife here are the first generation, right? But my children or offspring are the second generation. So it's from generation to generation. The halving effect to 23, right, of meiosis overcomes or neutralizes the doubling effect to 46 of fertilization, thus maintaining a constant chromosome number 46, right? From generation to the other, from parent to offspring. Now, the next part, we also have to know the last one, which is the last importance. Meiosis is a mechanism that introduces in genetic variation. So that's the third importance of meiosis, right? This happens through what? Crossing over. Remember, crossing over happens in prophase 1. So we said we are going to deal with this. Now, here it comes. So crossing over happens in prophase 1, right? That's where chromosomes pack themselves in pairs next to each other and join on the chromatids, right? And share genetic material. So that's your crossing over in prophase 1. And also random arrangement of chromosomes at the equator. This happens in metaphase 1 and 2, right? So that's your random arrangement of chromosomes at the equator. Now, the next part, we have to know your abnormal meiosis. So I'm just going to turn over the page. Okay, now the next part, abnormal meiosis. We have to know the causes and effects of abnormal meiosis. So those are the two parts we have to know: the cause of abnormal meiosis and the effect of abnormal meiosis. Now, I just want to remind you of these two things, right? Meiosis takes place in the testes and ovaries in humans, but in plants it happens in the anther. So please remember that. When you are dealing with meiosis, it happens in testes and ovaries in humans and the anther in plants. Now, here we go. Abnormal meiosis is caused by a non-disjunction. Okay, now a non-disjunction, right? This is a cause of abnormal meiosis. Now we have to know, when does a non-disjunction happen? So it happens during anaphase. It can either be anaphase 1 or anaphase 2. It can happen anywhere, in any one of those. So a non-disjunction is the failure of chromosomes to separate, right? It's when chromosomes fail to separate, right? Chromosomes fail to separate, right? Let me just add that, just to not to confuse people. So it's the failure of chromosomes to separate, right? Now, when a non-disjunction happens, it leads to the formation of an abnormal gamete. An abnormal gamete with 24 chromosomes. Remember, the half chromosome number is 23, but an abnormal gamete would have 24 chromosomes because it has an extra copy of chromosome number 21. So a non-disjunction affects chromosome number 21, right? Position number 21, right? Okay, now this is what happens when a non-disjunction takes place. So a cell, right? This is the original cell at the beginning of meiosis. Now, first division, meiotic division, just houses the chromosome number. It doesn't make the cell diploid. It just houses the chromosome number. Remember, first cell has 2 chromosomes, but the 2 first cells formed have 1 chromosome each. Now, a non-disjunction could have happened in this point, at this point. This cell would have had no chromosome, but this cell would have had 2 chromosomes still. But this time I decided to make it happen in meiosis 2, right? So meiosis 2, that's where haploid cells are formed, right? Haploid cells are formed. Now, in the second meiotic division, this chromosome was supposed to divide and be single chromosomes. So each single chromosome will be in each cell, but this time because of a non-disjunction at this point, right? A non-disjunction during anaphase 2, right? Anaphase 2. Now, so you have to be able to identify when the non-disjunction happened, right? So it happened during anaphase 2. Remember, this is meiosis 1, this is meiosis 2. Now, this chromosome has an extra copy. Can you see that? Has an extra copy at chromosome number 21. But this chromosome has 0 chromosomes because that chromosome that was supposed to be this side went to the other side. It was unable to separate from the other chromosome. So it was pulled to the opposite pole. And then to the opposite pole, nothing was pulled to that side. Now this side, the anaphase was normal. So as we can see, each cell has a chromosome, a haploid chromosome, right? So now you can also identify a non-disjunction or the effect of a non-disjunction, which is when an abnormal gamete fuses with a normal gamete, right? So that means it's 23 plus 24, right? Abnormal 24 chromosomes, normal 23 chromosomes. This will lead to a Down syndrome. It may lead to a Down syndrome. Now this whole thing may lead to Down syndrome. So this is how you identify that a non-disjunction took place on a karyotype. Remember, a karyotype shows the shape and size of your chromosomes from chromosome number 1 to chromosome number 23. Number 23 is your gonosomes, right? Your sex cells, gonosomes. So now at chromosome number 21, you realize that there are 3 chromosomes. Remember, these chromosomes have to be in pairs meaning in 2 by 2s, right? But this chromosome has 3 chromosomes, which means there is an extra chromosome. So that's how you identify a Down syndrome or non-disjunction on a karyotype, right? This is a karyotype. Now, this is all I have prepared for you guys. So now I need you to remember that the Down syndrome only happens when an abnormal gamete fuse with a normal gamete that may lead to a Down syndrome, because a zygote with 47 chromosomes will be formed, right? This zygote is abnormal, so that means this zygote may have Down syndrome. Okay, this is all guys I had for you on abnormal meiosis. Okay, thank you guys for tuning in. That's all I had for you. That was Life Sciences Meiosis. If you need more information on that, you can just WhatsApp on 076-587-5041 and you'll be added to a group which is Tutoring Wisdom. If it's not Tutoring Wisdom, it's not us. Now I'd like you to search us on YouTube on these channels, Lwazi Films or Tutoring Wisdom. Subscribe, like and share. That will be much appreciated. Thank you guys.